All right, so now I'm recording. So let's go ahead and go through the midterm from just start to finish, and we'll cover everything that you guys need to know. Now, for the very first lab we did the ubiquity lab, right?
Yeah.
In the ubiquity lab, you guys took samples of different things, door handles, clothing, maybe your shoe. If something's a non-living thing that can harbor or carry on a pathogen, what is the term for that?
It's not an endospore, is it?
No, endospores are totally different. An endospore is something that it's like that we'll get to an endospore a little bit later, but no, not quite. It's a fomite, if you remember that term. Yeah, so a fomite is basically any non-living thing that can harbor a pathogen that you can potentially transmit a disease to somebody, right? And it's a non-living thing. So, for instance, even food prep, if you had meat or chicken or beef or something like that, even though that used to be a living thing, right? Yeah. That could still be considered a fomite. So anything that's non-living. There's a term for infections that are spread through hospitals. What's the term for that?
Nosocomial.
Yeah, nosocomial infections, which can be markedly reduced by doing what? Hand washing. And then I also always mention, because I've worked in the UK for a little bit with the NHS, they had a nothing below the elbows policy, right? So they would have no rings, no long sleeve shirt. If you did, you'd roll it up. No white coats, which is kind of crazy, but now here's a question about culturing bacteria. So now if you start with a broth, or you have an agar or a slant agar in a test tube, and then you take that and use that to culture another sterile medium, like inoculation, right? What's the first thing you start with? What's the bacteria that you find in the broth? What's the term for that? Okay, so it's called a stock culture. Yeah, so if you have a stock culture, that's what you're starting with. It could be a monoculture or a mixed culture, right? It could have a single type of bacteria in it, or it could have multiple different types of bacteria in it. And then when you take that and then you do culturing, inoculation of sterile medium, what's that process called?
Subculturing.
Yeah, subculturing, exactly. Perfect. Now, if you have a mixed culture and you're trying to subculture from that, what's the best way to try to isolate those colonies on a mixed culture? You use it on a plate. Yeah, streak plate method, exactly. So you can thin out the colonies, and then you can have isolated different types of bacteria that are isolated. Here's a question about growth patterns. Sometimes if you see a lot of growth in a broth tube, it'll look you'll have a difficult time seeing through it, right? So that's turbid, right? So turbidity. Sometimes you'll see something growing in the middle-ish. Those are for microaerophiles. If it's a pedicle on the very top, that's going to be something that's aerobic. But if you have a little bit of sediment at the bottom, but it looks otherwise clear, what happened there? It didn't really grow. It didn't really take. It didn't really grow in the medium. That sediment is just bacteria that didn't really make it effectively. Yeah, so that means that there's no growth, really. Yeah, no growth. In terms of microscopy, what are the two most important things in microscopy?
Isn't that wave aperture?
Yeah, so that's going to be for resolution, right? So magnification is going to be one element, and then resolution is going to be the other element. And then resolution is going to be determined by wavelength, as well as what you said, the numerical aperture. The thing that you look into, the lens that you're looking into.
Is that the ocular?
Yeah, ocular lens, the one that rotates at the bottom. What's the one that rotates at the bottom? Objective, great. And then you know how to calculate, right? So just the true magnification is going to be the objective lens multiplied by the ocular lens. And the ocular lens is at set at 10, correct? Now resolving power, so that's resolution, is going to be determined by, it's going to be defined how? How do you define resolving power? So think about two objects that you're looking at. So the greater the resolution, the more defined the objects are going to be. So you're going to be able to determine between two separate objects, right? So in other words, the technical definition is going to be the minimum amount of distance between two objects, right? Where they can still appear as separate objects. So you can just pull up a seat. And then why do you use oil immersion?
Isn't it about the light of
Refractory index is going to be the same as what? Glass, yeah, exactly. Oh, yeah, absolutely. So, know the shapes and the shapes of bacteria and the aggregation patterns. You guys should know this by now. Yeah. So if it's round and in bundles and clusters, what's that called? Staphylococcus. If it's round and it's in chains, like a I don't know, like a pearl necklace. Streptococcus. Yeah, excellent. The way I like to think of streptococcus, think about strep throat, and then like one way to think about it, maybe it's like you're wearing like, you know, like a pearl necklace or something. Strep throat. I don't know. And then what's if it's a rod-like shape? What's that called? A bacillus. And if it's a rod shape in chains, streptobacillus. Yeah, that's great. If it looks like a corkscrew shape, what's that? Spirochete. Yeah, spirochete. Not to be confused with spirillum. Different organisms, but spirochetes, some of the major ones are going to be Treponema pallidum, which causes syphilis, Borrelia burgdorferi, which causes Lyme disease. So, know what your leukocytes look like. So first off, what are the 3 granulocytes? Yes. Yeah, so neutrophils, basophils, eosinophils. Which are the most abundant? Neutrophils, yeah. So what's the percentage range? It's like 50 to 70% on average. And then what are the 2 agranulocytes?
Monocytes?
Yeah, monocytes / macrophages. And then lymphocytes. What are the second most abundant of the leukocytes? Lymphocytes, yeah. So roughly 20 to 30% of your white blood cell count are going to be lymphocytes. What color are eosinophils versus basophils?
Purple? Is like the inside?
Yeah, the basophil looks kind of purplish, bluish, right? Eosinophils are going to be kind of reddish, pinkish. Neutrophils are going to have a multilobular nucleus, right? So if you see that nucleus that kind of has like sometimes 3 lobes, sometimes just 2 lobes, know that's a neutrophil. For basophils, it's kind of harder to see the nucleus. It's going to be covered in like those purplish granules. And what are those associated with? What are basophils associated with? Histamine, allergic reactions, right? So those plus mast cells are going to be associated with histamine release.
For eosinophils, you said it's like pink?
Kind of pinkish. They got kind of these pinkish granules. And those are going to be associated with what?
Parasitic?
Yeah, parasitic infections, exactly. Speaking of blood, so your blood is going to be made up of several different components, right? So you have your red blood cells, platelets, you have like 1% of your blood is actually going to be white blood cells, right? And then the rest is going to be plasma, right? So plasma is going to be all associated with like your humoral immune system, because recall that plasma is going to be differentiated B cell lymphocytes, and they're going to secrete immunoglobulins. Plasma is going to be mostly made up of water, but there's also going to be some other components as well. So what's like one of the major components in plasma? No, that's in red blood cells. Hemoglobin's heme, which makes up the oxygen-carrying component of red blood cells. So plasma cells are going to contain albumin. So plasma is going to contain albumin, which helps to maintain what? So if you were starved of protein and you had something like kwashiorkor, right, where you have 0 protein in your diet, you're starving to death, but you have like a distended belly, right? The distension of the belly is due to ascites and also fluid escaping from your vascular system and into the interstitial space, right? So that's what results in edema and swelling, ascites, things like that. That's because albumin helps to maintain osmotic pressure. So it allows for water reabsorption back into the bloodstream or the circulatory system. Albumin also helps to it's a carrier protein, so it helps to carry all sorts of different components like things like fatty acids and steroid hormones, for example.
So plasma only has water and albumin?
There's some other components too, but the albumin is one of the major components. There's like immunoglobulins in there. Yeah.
Okay.
So, but in terms of osmotic pressure and like the most important protein, it's going to be albumin. How do you fix a slide when you're preparing a smear from a solid medium? We've done that several times this semester, so you guys know it off the top of your head.
Like heat fixing and stuff like that?
Yeah, so what's the first step? You get the slide, right? What do you have to put on the slide first? Do you put the bacteria directly on the slide? Yeah, you put water on the slide, and you use a sterile inoculation loop to mix that bacteria in there, and then you do what? What's the next step? Do you heat fix it right away?
No.
You let air dry, and then you do the heat fixing.
So it's grab the slide, water on the slide?
Yeah, so if it's a solid medium, if it's a broth, you can just put the bacteria directly on the slide and then let it air dry and then heat fix. Mm-hmm. But if you're going from a solid medium, like a petri dish, like an agar, then you're going to want to put water on there first so you can mix it in. Staining. So after you prepare your slide, then you're going to stain, right? So we have a couple different stains. So we have simple stains, and then we have differential stains. What are the two simple stains that we've used? No. So simple stains, we like in our class we use methylene blue. You can use like safranin, crystal violet. I guess you could use crystal violet, but methylene blue and safranin are going to be the two most common ones. But what are those called? What's the electrical charge of those stains? Positive. So those are positive stains, right? So positive staining, you're allowing the stain to enter into the negatively charged cell, and then what does that allow you to do?
Allows the color to go in.
Well, what does it allow you to do? Like when you look at the slide now in microscopy, what do you see?
Oh, color.
You see the cell. You can see the shape of the cell. You can see the size of it. You can see the aggregation patterns of the cells. What about the other stains? So we used positive stains, and then we also used what? Negative stain, right? So negative stain, negatively charged. It gets repelled from the inside of the cell, so you can see the background really nicely. What does that allow you to do? What kind of organisms is it good for? Like a negative stain. You're not just seeing the shape of the cell, but you're also seeing some other components of the cell as well. So capsules? Yeah, so encapsulated organisms you'll be able to see with a negative stain. So that's good for that. You can also see biofilms too, but capsules are kind of what we were stressing in lecture. And then know that basic stains are going to be hold on a second. Let me see here. Yeah, so basic stains are going to be your positive stains. Negative stains are going to be your acidic stains are going to be your negative stains. All right, now speaking of capsules, what are capsules made of? What's the components? I'll give you a hint. It's sugars. Polysaccharides, glycocalyx. It's a polysaccharide glycocalyx. Like you can see polysaccharides also in biofilms, right? So biofilms are going to be kind of like a sticky goop that's released by the bacteria that allows them to adhere to surfaces and also to aggregate, right, and colonize the surface. Glycocalyx is going to be a little bit more of a harder material, right? It's a more dense type of like sugar, and it's going to allow for three major things. What does it help the bacteria do?
The capsule?
Yeah, what do capsules help bacteria do?
Oh, it protects it from getting.
Yeah, it helps them. It prevents phagocytosis. It also prevents antibodies from attaching to them. It also helps protect them against antibiotics, right? And so because it makes them more aggressive, what is it called? It starts with a V. It's a virulence factor, right? It contributes to their virulence or their ability to result in like pathology and disease. Now, when it comes to gram staining, we use crystal violet, right? We also use iodine. What's that iodine called technically in gram staining? It's a mordant, yeah, and the mordant allows what?
Doesn't it like help the stain?
Mm-hmm. Yeah.
Fix, like, the color?
Exactly. It helps that primary stain remain fixed within a gram-positive organism. So it helps it get held more tightly into the cell. Now, when you do gram staining, what's going to be like the ultimate goal of gram staining in a clinical basis?
Differentiate between like
Yeah, so you see like gram positive versus gram negative, right? And that helps you do what? Determine what kind of antibiotics to use, right? So it helps you determine like how to target the bacteria with antibiotics. Like gram positives are going to be by and large more susceptible to things like beta-lactam drugs, as well as like vancomycin, which is a similar mechanism of action to beta-lactams. It targets the peptidoglycan cell wall, right? I have a picture. Remember the image of the guy with the pistotonus, where his back is arched. He's like hyper flex. He's rigid paralysis, spastic paralysis. It's caused by what? What bacteria causes that? Tetanus, right? So Clostridium tetani. And what's going to be one of the aspects of Clostridium tetani that contributes to its virulence?
the inhibitory. Oh, I just have this.
Well, not the tetanospasmin, but the actual component of the bacteria. It can survive in like the environment for like up to 100 years. No, what allows it to do that? What allows it to do that?
Dormant.
Yeah, the dormant. What's the dormant stage called? It's a spore.
Spore.
Yeah, so it's a spore. It's a spore-forming bacteria. So Clostridium tetani, Clostridium botulinum, what others? Clostridium difficile. Those are all spore-forming. Anthraxis bacillus. Bacillus anthracis, Bacillus cereus. Those are all spore-forming bacteria. So those spores are going to be able to survive a very long time in harsh environmental conditions. And then, are those going to be reproductive? No, they're in hibernation, right? So once they get introduced into a more optimal environment, then they become a vegetative cell. And once they're a vegetative cell, then they can actually start reproducing and undergoing just normal metabolic activity. Now, we also did acid-fast staining, right? Remember, we used like carbofusion, and we also, did a counterstain with methylene blue. What are the acid-fast components? What makes an organism an acid-fast organism?
Is it the
Mycolic acid, exactly. And what are some of the major infectious agents that contain mycolic acid? So Mycobacterium tuberculosis, Mycobacterium leprae. Those are the main ones that we focused on. The ones that we stained in class were Mycobacterium smegmatis, but that's neither here nor there. Tuberculosis and leprosy are the two major ones that we're concerned with. What's tuberculosis associated with in terms of like social conditions, environmental conditions?
big crowds.
Huh?
Big crowds.
Yeah, crowds, exactly. So congested areas, overcrowding, overpopulated areas, poor sanitation, right? Poor ventilation, things like that. Speaking of environmental conditions, what are like the 4 major environmental conditions that contribute to a bacteria's ability to survive? One of them is temperature. pH, oxygen, water. Great, exactly.
What was it? Environmental.
They're like environmental factors that contribute to the bacteria's survivability. Right. So some bacteria like it cold, some bacteria like it hot, right? So is it a psychrophile versus an extremophile versus a thermophile, right? Some prefer no oxygen, right? So obligate anaerobes don't want oxygen, right? Clostridium tetani is a good example of that. Once it gets exposed to oxygen, it starts forming spores to protect the DNA from oxidative stress. Some like low pH, some like higher pH, right? So Helicobacter pylori is a really great example. It lives in your stomach, right? It causes stomach ulcers. It doesn't like low pH. It actually doesn't like the acidic environment of the stomach. So what it does, it starts releasing things like ammonia and starts creating a more basic environment. And that's what compromises the stomach lining, resulting in stomach ulcers. So all those factors are going to be important for bacteria growth. What does a lymphocyte look like? Going back to your leukocytes. So remember the neutrophil, multilobular nucleus, eosinophil, kind of pinkish granules, basophils, kind of purplish, bluish granules. Basophil blue. Think basophil blue. Monocytes are going to be kind of really big, right? They're kind of like 5 times the size of an RBC. They have like a horseshoe / kidney bean shaped nucleus. Then what does a lymphocyte look like?
Isn't it like basically the whole thing? Or
It's
like-
the nucleus is almost the entire cell, right? And then you get a little bit of cytoplasm off to the side, right? So kind of like a crescent-shaped cytoplasm, and it's going to be like almost the size of an RBC, sometimes about twice the size of an RBC. What kind of infections are lymphocytes going to be associated with predominantly?
Or what?
What type of infections are lymphocytes associated with? Lots of different things. Viral infections, bacterial infections as well. Also, they have anti-tumor properties, right? So like cytotoxic T cells, they will be able to identify cancerous cells, and they'll induce apoptosis using tumor necrosis factor as one of their cytokines. So they're not going to be really associated with allergies, and they're not going to be associated with parasitic infections. It's mostly going to be viral, bacterial, and cancer. Can you tell the difference between a T cell and a B cell lymphocyte? They look exactly the same. Now, in terms of antibiotics, there's going to be 2 major categories of antibiotics. Ones that kill the bacteria and ones that prevent the bacteria from growing. What are those? What's those terms?
Bactericidal.
Yeah,
bactericidal.
exactly. Bactericidal, think homicide, right? Bactericide, killing the bacteria, versus bacteriostatic, which is prevents the growth. And then you're relying basically on the host's not only immune system, but also mechanical function to be able to remove the bacteria. So like if you use something like, I don't know, trimethoprim sulfamethoxazole, which is a bacteriostatic compound for like a UTI, then you're preventing the growth, but you're depending on the patient's ability to pee the bacteria out. It's just mechanically flushing the infectious agent away. Now, in terms of antibiotic resistance, what are the 4 major mechanisms that bacteria can develop resistance? Like in the way that bacteria evolve resistance capabilities. I'll give you one hint. Blocking the entry of the drug, right? So it literally just repels the drug, doesn't allow it to come in. That's another way.
Yeah.
That's called a what? There's a term for that. So that you can block the entry, or they can have efflux pumps. So the moment the drug enters the cell, it just pops it right back out. It's like, someone throws a grenade at you, you say no, and you throw the grenade right back at them, right? It's kind of a way to think about it. Then there's 2 more ways. What are the 2 other ways?
Takes it in and it like adapts to it or something like that?
Break it down, actually. How do you break something down?
Dissolve it?
Huh?
Dissolve it?
With enzymes. So think about like beta-lactams. If a bacteria has a beta-lactamase enzyme, then it can break down the bacteria through enzymatic degradation. And then what's the last one? So back to beta-lactams, there's going to be a component in the bacterial cells called a penicillin binding protein. That's where some beta-lactams bind to. If you alter the penicillin binding protein in the bacteria, the penicillin has nowhere to bind to. The beta-lactam is going to be rendered ineffective. So those are the 4 major ways, okay?
So block, efflux.
Yeah, so blocking efflux pumps, enzymatic degradation, and altering the final binding protein, or the binding site of the drug. And then how do bacteria divide? Binary fission. Excellent. Awesome. That is it. You guys have any other questions?
So that's all the stuff that would be like from the midterm?
Yeah. Yeah. I'm taking 25 questions of those and I'm putting them on the final exam. I may or may not do some alterations on there, but probably not.
So it'll be those 25, the stuff that was in the other video?
Yes, exactly. And I'm going to post this. I'm going to make a video of this as well for you guys. So, all right, cool. Thanks for joining me.
Thanks.
All right, no problem.